morning. Welcome to this 10 hours course on psychiatry and overview. We have tried to divide this course into 4 modules, and in the first week we will be focusing on the basis of human behavior. But before we move on to the basis of human behavior, let us clear a confusion. In common public and lay people, there is a whole lot of confusion between what is psychiatry and what is psychology. To give you a brief definition, psychiatry is the branch of medicine which is supposed, and it does, deal with human behavior and its abnormalities, essentially trying to understand the illnesses arising out of mind, the behavioral disorders, and attempts to treat it. So, people do their medical graduation and then they specialize in psychiatry, whereas psychology, which is obviously a older discipline, is a study of human behavior in general and its application in education, organization, various other psychological concepts of mind. We will discover this interface as we move on. Psychiatry also interfaces with other branches of medicine like neurology and general medicine and has its implication in that, but all that we will cover in the coming few weeks. For now, we will try to understand why do human beings do what they do. So, this understanding of human behavior has a basic question: how do the thought, emotion and behavior arise from this portion of our body called brain? As we go on in this 4-5 hour, 2-3 hours of lectures in this week, lot of this is connected because they are all connected to the human brain. So, a lot of information may overlap or one lecture may telescope into each other. So, please try to understand it as a composite whole. There are no clear cut division in human behavior, and so is in the brain. But to start from the basic, this structure of human body called brain is 1.3 kg of almost pinkish structure safely enclosed in your skull, but its activity creates you, it creates what we do, it creates all the human interaction, society and all that goes on with human life. For practical purposes, and there is a confusion and there is a lot of philosophical debate on what is mind and what is brain, but for our purpose all through this lecture we will use it interchangeably. And for a common understanding, as a very simplistic statement, I would like to tell and that mind is what the brain does. As the brain goes on with its functions, mind is created, and mind is a composite whole of the behavior, the interactions, the feelings, the emotion. So, let us go over this basic anatomy and physiology. Both are interrelated in brain, as history and geography both affect each other in a reciprocal function. So does the anatomy and physiology in human brain. If you grossly look at the brain, what you will find is it has the front, which is called a frontal lobe, the parietal lobe behind it, on the sides a temporal lobe, and there is a occipital lobe at the back, and tucked in between is the midbrain, and below it is cerebellum. And this portion, this white portion which you see, is the medulla, which continues as the spine and a spinal cord which is safely tucked into the vertebral column. They form the part of spinal cord, and the brain form a part of central nervous system. We will not be bothered too much about the spinal cord, we will focus on the brain because that is what we are concerned with in psychiatry. Although the peripheral systems are of interest to us, but mainly it is the behavior. So, we will focus on the brain and as a part of central nervous system. Let us go back to history, because we did not know always that the brain is the seat of thinking. So, there have been lot of philosophical mulling over this issue. Initially it was thought, like poets still think, that is the heart is the important thing, and so because the emotions were always predominant and they are still predominant in most people, they form a major spine of behavior. So, it was initially thought it is the heart which is the center of thinking. As thinking progressed, then we evolved, we thought it is the ventricles, those small areas in the brain which are the site of thinking. But now we are sufficiently sure that through advancement in knowledge, thought and technology, that it is the brain eventually which is the center of thinking and feeling. So, heart may rule the poetry, but eventually the emotions also lie in the brain. This is one of the images from a functional MRI, and these are the areas which are demarcated. We will talk about functional MRI later, where this is the area where we hear words, just for your preliminary introduction. What really got a fillip and a impetus to thinking was this something called phrenology, which is no more considered as a science, but phrenology was brought by a person called Gall in early 1800s, and he emphasized that the brain is the organ of the mind. It is composed of multiple distinct innate faculties, and because they are distinct, each one of them, each faculty must have a separate seat. So, what he was essentially saying, that each behavior or a component of behavior has a separate seat, and he really wanted to check all this through the bumps on the forehead. Right. This at that time probably became very popular, but it is no more considered as a sign. But to give the devil its share, it may not be the bumps on the forehead which may be telling this, but more or less through the modern technology and by the various tools of studying brain, we have this accumulated this knowledge over years, where we can demarcate grossly the areas from which the functions are regulated. For example, if you see this area, yellow area, where it is for the motor preparation, like this area in the frontal cortex is for the anticipation. This area is for hearing, mathematical calculations, and this underlying area is for smelling. This area is for vision, the movement, the shape of the objects, and so on and so forth. If we look at this area, if we divide it from in between, the areas in the front is responsible for movement, the area behind it in the parietal lobe is responsible for perceiving, for the sensations, pain, temperature, touch, and the whole body is represented in this parietal area, with foot on the top having the maximum area of representation, and then so on and so forth. So, any peripheral sensation which arises from the body goes through the nerves to this area, where it is integrated in perception, and if the signal is passed, we will discover this as we talk about physiology, and the brain decides to act on it and sends a signal to the spinal cord, obviously modulated by cerebellum and other areas, to respond to it. That is what happens in a reflex action. So, if you suddenly touch a hot thing, your hand recedes back. So, within that milliseconds, 100 of milliseconds, the signal goes to the parietal lobe, the heat is felt here, the signal is passed, and the brain directs the hand to recede. So, phrenology may not be true, but at least in some way or the other, within the brain with technology, we know that there are certain areas which are specialized for certain functions. The microstructure of the brain was really discovered by these two people, Casal and Golgi, where they discovered that the brain is really made of these fiber like things which form a network. So, neurons are the units which form the neural network. Perkinse, Ghazal, Golgi, they formulated it. The further discovery went on that how are these neurons connected. Neurons are like fibers, they are like cables which are running in the brain and in the magnitude of 10 to the power 10 or 10 to the power 11, and they are connected to each other through something called synapse. We will discover this synapse as we move on. This guy Fiennes Gage was a person who had an injury with some solid iron rod. He survived, it just pierced through his left frontal lobe, and what was found that his behavior dramatically changed after this injury. That was one more idea which came to the researchers at that time, that an injury to the brain can change your behavior, and using a reverse logic, it was inferred that maybe this certain behavior which was happening before the injury is controlled by that area. Now, this became one of the ways of studying the brain. So, now if I go back and see how do we study, what are the approaches? So, the approaches to study the brain are like anatomy, physiology, where we trace the track, we do single unit recordings, behavioral studies, which is a part of psychology, patients with brain lesions, people who have damage to the brain by illness or by injury, the behavioral changes which they undergo are correlated with the site of injury and the difference in function it gives rise to. Postmortem anatomy, this was one of the very popular ways that obviously we can't go into a live brain very deep, and so once we do postmortem and then correlate what was happening during the lifetime, gave us an idea of what was happening. Structural imaging, which is a new technology we will be talking about later, neuroimaging and brain mapping. So, in the last 40 years, we have learnt about brain more than what we did in the last 3000 years, all due to technology, but this technology is also based on what behavioral observations and anatomical lesion studies were done in the last 60-70 years of between 1900 until late. Penfield was a surgeon who could find out areas of brain leading to certain functions, motor functions or sensory functions by stimulating those areas. During neurosurgery, he used to stimulate certain areas and see what was happening in the body. Either the limbs were moving or certain sensations were felt. This gave a huge, it actually opened a wide window of understanding the brain. In localizing of functions, along with Penfield, Paul Broca, when he studied the brain of few people who had lost language and speech after a stroke, he found out this Broca's areas which is on the left side, tucked in the left side of the frontal lobe in the dominant hemisphere. So, what was discovered that language area is in the left hemisphere. In most right handed people, it is the left area, and one thing which also came out with research was that the left area controls the right motor area of the body, and the right side of the brain controls the left side. And language is a function in most people is in the left dominant hemisphere in the frontal lobe. Almost following it was the people who were able to speak, but were not able to comprehend the language, and Wernicke's receptive aphasia is a term used for it, which is in the temporal lobe. So, now we knew that in the frontal lobe damage causes Broca's aphasia, where people are not able to express in language. In the side of the temporal lobe, people here, if you have a damage by a stroke or by tumor or injury, people are unable to comprehend speech. But this was still not clear, that why these two separate areas, how do they connect, how does speech connect. So, the third big thing was something called discovery of a conduction aphasia. where Broca's area was intact, the Wernicke's area was intact, but in between the damage caused conduction aphasia. The person could express, the person could hear, but could not relate the two things. This gave the idea that brain may actually be working in a network. We will discover those networks as we talk. Again, to revise it, this is the gross structure of brain. Frontal lobe, where I said on the left side is Broca's area. Temporal lobe, this area is Wernicke's, where you hear. Parietal lobe is all sensations go in here. This area controls the motor movement. And occipital lobe is for vision. Cerebellum is for fine tuning of motor movements, of body movements. So, this is how the brain looks like. And if we cut the brain, it is something like this. This is the cortex, what we call, we all have heard of gray matter and white matter. This colored area is all gray matter. Actually, it is not gray. A human live brain looks like pink, and this is all white matter. This white matter forms the nerves, actually, the neurons, it comes down. So, all this, if you look at this, the gray matter is composed of neuron cell bodies and some branches coming out of it called dendrites. And this is the cortex, is a 6-layer structure which covers the whole brain. White matter, these nerves are covered with an additional sheet of fat, and which come down and carry the signal to different areas of brain and down to the spinal cord. This area is the midbrain. So, we have a outer layer of cortex divided into frontal and parietal and temporal. Within this cortical layers is stuck the deep areas of brain. If we keep slicing, we will see different things. These are all basal ganglia, which are form of a part of midbrain. So, brain is also not a composite one single structure. It also is divided into two hemispheres. So, if you look at it now, we have a brain which developed in the last millions of years after we diverted from chimpanzees. The brain has evolved to have an older structure which is responsible for movement, which is present in lower species of animal also. It has a structure called midbrain, which came somewhere in between during evolution, and the neocortex. We have a paleocortex, we have archicortex, and we have a neocortex. The neocortex is the latest structure which evolved, which is responsible for our thinking and all the complexity of human brain. So, brain has two hemispheres, cerebral hemispheres, left and right. You would have heard of right minded and right brained and left brain. This is partly as we have understood the behavior and its areas which control the behavior into, they are specialized into, some of them are well done by left and some by right, but they do not function independently, though they have independent functions, but they are connected by this structure called corpus callosum. This whole corpus callosum is a structure which, if you can see this, the arrow, it is here. It has maximum number of neurons and connects both the hemispheres. It goes on like this. The cortex of both the hemisphere is connected. So, brain is actually taking in information, integrating it between two hemispheres and leading to some action on it. So, if you cut the brain, from the side, from between the two hemispheres, this is the type of networks and fibers which you will see. So, this is within one cerebral hemisphere. So, it is connected from front to back, up to down, and with corpus callosum between the hemispheres. This is just a broad gross thing to show you how we comprehend and we generate the words using various inputs, right. So, like you take this visual thing, from here the sensations go, the word go here, and is the frontal lobe which gives the meaning to it. auditory, it goes to the temporal lobe and it goes to the frontal area where the meaning is given to it. Here it is like a somatosensory, touch is given meaning to this. So, they are, as I said, the temporal lobe in a very gross terms is the temporal lobe where you hear, parietal lobe you feel, and occipital where you see, but all this information actually is circulated through the network, and it is the frontal lobe where the meaning is given to it. We will discover this as we go around studying the physiology. This again, as I said, these are the images from functional MRI. This is a passively viewing words, when you are just seeing the word and not thinking about it. When you are listening to the word, this is the temporal area again. When you are speaking, these areas get active, and this is what you are generating words. Generating words is in the frontal lobe. Now, as I said, if you see the broadly, this is a sensory motor area, there is a cut in between. Here the sensations come, foot is on the top and rest of the areas. This information, here the words come, here it is a visual thing. There are also these areas are present in all the primates, but why human brain is different is with the development of association areas. Association areas are more specialized area which are used to coordinate and integrate information, like this is the auditory association area in addition to the area where you listen word. This gives meaning to it, and this includes the Wernicke's areas, like you find the frontal lobe where words are generated, Broca's areas, but there is also, this is a frontal area. We also have prefrontal area. Prefrontal area is considered to be the chief executive where decisions are made. Prefrontal area assesses on what action to be taken, the salience of the importance of the stimulus which comes. We will talk about these networks as we move on. So this is it's like a precentral gyrus. If you see this cortex, you can see a lot of these folds. So the valley between the folds is called sulcus, and these folds are called gyri. If you see it here, it goes like this. It almost appears as a fractal geometry. This is called a gyri, and this is called a sulci. This is a very smart trick of nature to increase the brain area, to fit in 10 to the power 10 neurons, because otherwise if you spread them, you can imagine how much space it would take. And this is the gross structure. These are cerebral peduncles, these are the midbrain, the neurons come from here and they go on to spine. So, this is the motor system again. So, motor system also is included here, where because there is a structure called basal ganglia here. Basal ganglia and cerebellum actually fine tune the movement. Initiation of movement, decision making on the movement is done in basal ganglia. So, any damage here can really lead to, you would have heard of an illness called Parkinson's. It is actually a problem in one of the areas called substantia nigra, where a deficit in one of the chemicals and damage to this substantia nigra causes difficulty in walking, slow steps and tremors. Similarly, cerebellum, which is stuck here, this structure is also responsible for fine tuning and whatever feedback loops come to the brain regarding your orientation in space and time and your body, the movement of the body. This is how again the motor system goes. Right. So, this is again like the neurons start from here, go to the internal capsule of basal ganglia, go to the spine, midbrain, again a structure in the brain, pons, medulla, and then to the spinal cord. From spinal cord, the synapse with motor neurons which goes to the muscle. This is a typical structure of what neuron is made of. There is a cell body. There are branches like this, multiple branches like this called dendrites. This cell body continues into the axon, and axon ends here, but it terminates on the dendrites of the next axon, and which has another axon. So, it will go again and turn on the dendrite of something else. So, this is one structure. if you look at it, and this area is the synapse. So, now you can understand almost 10 to the power 10 neurons in various areas of brain form a huge network. Each of this neuron is a body in itself, which has a nucleus. Dendrites are the receiving end and axon is the passing end. So, dendrites receive information and it is all about passing information. The brain is just receiving information from the environment, integrating it, passing it between neurons and through the network, and finally taking an action or a thought or emotion out of it to keep you interfaced with the world. So, dendrites take the information, pass on to the axon, then it ends here. That there is a gap between the dendrites and the axon, this is a space here. Now, this space is managed through chemicals. We will talk about it when we talk about the physiology. Again, this is one of the, so this is what I was telling you, this is how in the parietal lobe the body is represented. On the top is foot, and you can recognize your own organs. This is little ring, middle index finger. Hand has a huge representation as compared to the other thing. Face has a huge representation. And this is all has happened, nobody has sat down and planned it. It is more a evolutionary step which the brain kept learning as it interacted and as it turned from reptile to you and me. I told you about the basal ganglia, which is tucked here, and this is responsible for a lot of fine tuning of movement. And this is cerebellum. If you just take a part of it, it appears like this, and this is a different structure. So, again, as you saw the motor thing coming down from here to the muscle, this is how it goes up from there. If somebody touches you, the signal goes down to the spinal cord, goes up to this structure called thalamus. Thalamus is an important structure in midbrain because thalamus is the relay center of the brain. On both sides in the midbrain, almost intermeshed with basal ganglia, all sensations reach thalamus and thalamus sends it to the cortex, where you feel the sensation. But all sensations have to pass through thalamus, even vision and hearing and touch, because this is where all sensations are integrated and given a composite view. Because imagine if all other sensations are flying in 5 or 10 directions, it will be very difficult for the brain or for us for that matter to make a sense of what is happening in the world. Even while I am talking to you, there may be 1,000 sensations going on in your body and my body, but brain is making a composite sense of it to give me a feeling of being one and as a unity. Auditory functions we have already mentioned briefly. We will skip this. The vision is very special thing. A lot of information about brain has come from vision. This is the area of occipital lobe from where we see. So, we have 2 eyes, this, you see these areas. If you divide it, draw an imaginary line, there is a left optical field and there is a right field. So, data from this, both the lefts, they go down through the optic nerve, go to the retina, they come down. Half the fibers come like this, half the fibers cross. So, eventually what is happening is, if you look at it, the signal from this and this will eventually come to the left side. Signal from right will come to this side. So, the left field goes to one side, the right to the other side. This is for the binocular vision. Otherwise, we would not be able to see the, it is like you are taking signal from 2D and you are seeing in 3D. Although it is a 2D vision, so and that is why you feel differently when you are seeing it in a 3D, but brain has to create the image of the objects. It is like creating 2 copies for us to better comprehend it in a 3 dimension. You can see this further. So, right hemifield is going here and the left is going here through crossing. You can just track the fibers, and finally, it goes to both the sides to create a. So, light goes in here. This is like photons go here, go to retina, bind with some certain pigments there, change the shape of this, which sets in an electrical current, which goes through the optic nerve. Optic nerve goes here, half of them cross here. Before that, they go to what you call a lateral geniculate nucleus, which is a part of thalamus, as I said. So, from here it goes to occipital lobe, and occipital lobe has these layers of cells. Now, these layer from 1 to 4 are specialized to capture movement, to capture shape, to capture the contrast. So, if you just for a second look at the whole thing and see how wonderfully nature has really made it. Let me go back to some signal goes in from here in the form of photons, which is electromagnetic radiation, goes to retina, photons bind, set in an electrical current, and this electrical. So, there are the multiple of them. So, all the photons are going, and this electrical current divides into these neurons, goes to the midbrain where the integration of signal happens, and then it goes to the occipital lobe from layer 1 to 6. The various feature extraction is done just by interpreting the electrical current, and an image is formed. This image again is transmitted to the frontal lobe. In the process, it is compared with the existing memory or map of the image in the brain, which this area does. And then a composite image comes to your brain, on which the basis of which the brain decides whether it wants to act on it, it wants to ignore it, and this is how the brain keeps you in touch with reality. So, this amount of specialization of cells has evolved over millions of years. Not each layer does the same thing, not the same set of cells sees the whole thing. Some part of the layer is seeing the movement, some part is seeing the color, some part is seeing the contrast, and that whole information is put together to form a composite image of what you are seeing, which again is compared with the existing, if there is existing memory or map of that object, and both of them are compared to give another composite image to the frontal lobe, where the prefrontal cortex decides, and the other networks which I will talk about decide on what to do with it. And this is how the mind or the brain keeps you safe. Imagine a lion attacking you. Now, whatever light is reflected from lion will go into your eyes, an image of lion will be formed. This image of lion will be sent to your midbrain, where your midbrain will look into the memory of lion and decide that this animal is dangerous, and then it will send to prefrontal cortex, where it will decide to make you run, because if it fails, you are dead, you will not be there to look at another lion. And all this happens in, say, a period of, say, some 10 milliseconds to, say, 500 milliseconds. Okay. So, now we know the brain is composed of discrete cells, neurons, as I told you, and there are some things called glial cells, which are much more than neurons, and they are widespread in the brain between neurons, and they have different functions like packing, being used as a packing material, like being used as scavengers in case of infection. One of them actually also buffers the sodium potassium electrolyte level. So, the brain is composed of the 6 layers, which vary from area to area. The first 1 or 2 layers are mostly composed of cell bodies and dendrite, as you can see in this image, dendrites, cell body, axon. This is taken from here, but the layer 5th and 6th are the most important, because these are called pyramidal cells. The pyramidal cells are obviously the word is self explanatory, and the layer 6. So, in the layer 4 all the inputs from thalamus come. This is the fifth layer, fifth is from where all the commands go, the motor command and connections to the other areas of the brain to the spinal cord, and layer 6 is the feedback from the cortex to the thalamus again. Now, these layers may vary in different areas depending on the function of the area. Like in some areas, there are only 3 layers which are more developed, in some areas there are more 4, depending on what function that areas. But all these neurons, if you look at this histochemistry, they are arranged in the form of columns. So, there are micro columns and there are macro columns. There are no clear division. It is not as if it has been put in an envelope or a cylinder, but they are arranged in the form of a column when you look at it. Functionally, it may not be necessary that all columns should fire together on a certain action. The neurons which fire together can be separated far away from each other. How they function will just come to it. This is how it looks. At a microscopic level, these are the columns. Increase the scale, it is like this. And so there are multiple, each of these micro column are fitting into these macro columns. If you want to look at the scale, it is like this. Mini column is 0.03 and the scale of 10 to the power 2. These are mostly inhibitory connections. Cortico within the cortex, if the connections go like 0.3 and 10 to the power 4, macro column is almost 3, this is millimeters, and 10 to the power 6. So, see the increase, So, number of mini columns which can go into this, and as you go into the region which are largely called Brodmann areas, functional Brodmann areas, as I showed you that okay, this is the area for hearing, this is the area, they are called Brodmann areas. Each lobe has, this is the scale, and 10 to the power 10, and each hemisphere has 10 to the power 11, that is half the brain. Each lobe means frontal lobe and parietal lobe, hemisphere. So, see the increase in the scale in millimeters and the increase in number of the neurons, and as it is seen grossly. These are the Brodmann areas which have been numbered like 21, 42, 44, 44 is Broca's areas, so on and so forth. So, these are broadly divided according to the function. But does it mean that all the columns, all the cells in one column like this, all have to fire together, or all the areas which are specialized will fire together as and when the action arises? It has been found that it is not so. It is in fact the neurons may be sitting somewhere else, but they may be firing in coordination or in conjunction with neurons sitting somewhere else. It was almost like neurons which fire together, they wire together. So, wiring together does not mean that they have to be in the same geographical location in the brain. They can be sitting, but their action may be determined by the firing of some other neuron sitting somewhere else. Now, this is like from the graph theory. If you make a graph of how the neurons connect, so they are called Hebbian assembly. They modify their strengthening of the connection between the neurons is when they become active together. Like for example, in say, let me give you an example. In the case of memory, if certain memory has to be activated, all those neurons which were involved in forming that memory will fire together. So, they may not be necessarily in the same place, they may be scattered in different areas. Like for example, if you say a word rose, some of you may smell rose, some of you may see the color, some of you may see the petal, thorn, touch, anything can come. So, it is very unlikely that there will be a block sitting in the brain where it will be all about rose. No, the whole memory will be scattered, but one point as it triggers, all this will get activated. They are Hebbian assemblies. Hippocampus is the area in the brain deeply tucked in temporal lobe, which is responsible for memory mostly in forming memories, because the way it fires, increases the synaptic strength, which is called potentiation in the case of memory. And the other areas which have motor memory, the other areas which have olfactory memories. So, there are largely 5 big networks which function as forming and connecting all this. One, you can just remember the names, but look at it: parieto-frontal network for spatial attention. So, it is all about orienting in time and space. So, parietal lobe, where you get feedback from the body, all the sensation from the touch on the earth, your orientation, your standing, and which passes the message and integrates with the frontal lobe, which controls your body tone, body movement, and keeps you oriented in the space. Occipitotemporal from the back and temporal for face recognition. Temporal lobe and limbic system. Limbic system is the emotional network which is tucked within the brain, within the cortex, for learning and memory. Prefrontal network for attention and compartment. So, largely if you broadly if you look at them, you can call them 3 networks. One is called a default mode network, which is the neurons which are active at rest. There is a central executive network, the prime area of which is dorsolateral prefrontal cortex and cingulate gyrus, and there is a salience network. The salience network also looks at the feature extraction, looks at the important thing on what you are seeing and what you are reacting to. So, all these 3-4 networks actually keep you what you are. The mind through these networks always tries to keep you in touch with reality, and at the same time managing the deeper emotions and your thought and whatever is being internally generated at the same time. But the larger purpose of this one is survival, because if you are not in touch with reality, survival may become difficult. So, first and foremost is survival, and taking actions and doing motor movements and feeling sensations and feeling emotions and thought, which will keep you in the best fit in your environment. That is one concept of normalcy also, so that you are best fit in your environment. So, all that normalcy comes from this firing of networks, and then when a stimulus is presented, to take that stimulus in, to analyze it, to create an image of it, to integrate in whatever existing memory and maps you have, and then presenting it to the brain to take action on it, which may be most apt. Memory, which is formed through whatever stimulus is presented, if your brain keeps firing on that for some time, it will become a memory. Sometimes the sudden memory, the sudden impulse which may be very, very intense, can alter this. So, these are like synapses which are made between neurons, and as I said, if they fire together, they wire together. So, broadly I have not tried to get into the deeper things of anatomy, but this is how largely the brain functions. It takes in, creates an image, compares it with the existing memory, presents it to the higher centers, which take a decision on whether to act on it or save it or emote on it. So, as I said, the glias are the supportive cells which are. How does it happen? It happens through all this. Once the brain gets active and its electrical activity is on, it never stops till somebody really falls ill or dies, and brain death is the death actually. So, it keeps firing, and as you had seen in earlier slides, each neuron connects to the other neuron through dendrites and the end of the axon. There is a gap, as I said, synapse. What happens between the synapse? So, it is an electrical activity which is going on in the brain between these 10 to the power 11 neurons, and each neuron is connected to the other, or many, like on a dendrite, many axons may descend. But how do they cover that small gap which is a non-contact area, which is called a synapse? The electrical signal changes to chemical signal there. So, ultimately brain functions through electrochemical signal. Now, what axon So, if you see it here, this is a synaptic cleft. This is a gap, this is a gap. So, each dendrite here would be receiving, each dendrite here, this is a dendrite, each dendrite here has the axon coming from another neuron, and this is the synaptic cleft. If you see it, now this synaptic cleft, how does it bridge? This is the electrical activity flowing on here, and this is the myelin sheet with glial cells made. This myelin sheet actually makes the speed faster. Current action potential, we will talk about it in just a while. It goes on like this, it comes here. Now, how does this axon transfer this? This is through the chemical, which is called a neurotransmitter. If you look at this here, this is a myelin sheet, this is the axon, the current is coming here, and it is secreted into space. The dendrite, the other neuron, takes these chemicals and another set of electrical activity goes into it. Look at it, it comes on here, and these are receptors, these are neurotransmitters secreted from here. It is just a cartoon. It comes here. This is the whole basis of functioning of brain. Now, there are many neurotransmitters. Each set of neuron specializes in secreting one type of neurotransmitter, and they have a concentration in certain areas of brain, but the receptors are there for lot of them. The dendrites have receptors for lot of them. It's like, for example, this is a dopamine system. Dopamine is one of the neurotransmitters. These areas are where it is maximally present. This is noradrenergic. So, these are the maximally concentrated cell bodies of this, and then they have a wide spread presence all over. This is a serotonergic thing. You see, maximally concentrated here in raphe nucleus, like this is in locus ceruleus, this is in substantia nigra. This is where actually the damage happens in Parkinson's disease. Now, what is happening with these neurons? See, the brain functions in with something called a action potential. Action potential is that in normal condition, the outside of the brain is positive and this is negative, with a difference of say minus 70 millivolts. When there is a signal which comes like this chemical goes, this neurotransmitter goes, binds to this protein structure called receptor, and it goes in, it either excites or inhibits. Excite means it will set in a current, or it may be inhibitory, that it may not set in a current. So, that is simply managed in the, it's simple to say, but it is a very difficult process, is that if there is a high level of sodium outside and potassium and chloride inside, this will be positive and this will be negative, outside positive, inside negative. This is polarized state, but if a current comes, suppose this binds here, suddenly this will open, this channel which is normally closed will open, lot of sodium will rush in. This will become negative and this will become positive. The outside becomes negative and inside becomes positive. So, this is a depolarized state. So, this set of current goes on like a wave, and this is called action potential. Action potential is all or none, either the neuron is fired or it is not, it can't go partial, but depending on, so once it passes, the current passes through the whole neuron and it comes here, again this will decide on what is being secreted. Now, glutamate is one of the excitatory, so it will always go to the next neuron and excite it. That means it will set in a current. like there is something called GABA. GABA always goes and inhibit, that is one of the inhibitory thing. Now, what is the importance of excitation? And imagine this we are just talking about one neuron, but does not happen with one neuron. When it is happening, it is happening with millions of neurons which are secreting on one cell. So, if millions of neurons are sending GABA, that neuron, the next neuron is not going to fire. Inhibition is important because inhibition controls the activity, because if it is all excitation, all the neurons will keep firing, and you can imagine the confusion it can cause and the conundrum which it will cause, because brain has to regulate activity by controlling it. So, inhibitory neurons can also act in two ways. One, that it will go and inhibit the firing of the other neuron. Sometimes they can inhibit the one neuron, which in turn will excite the other one. Like, let me give you an example. Like, if you say there is a neuron 1, there is a neuron 1 and there is a neuron 2, there is a neuron 3. Now, neuron 2 inhibits the firing of neuron 3. So, it does not allow neuron 3 to fire. So, that means the functions controlled by neuron 3 will not be working when neuron 2 is active. But if neuron 1 inhibits the neuron 2, the control of 2 over 3 will vanish for the time being, and then 3 will start firing. So, it is like a loop which goes on. One neuron controls the other, it may get inhibited in turn by the other, and the functions controlled by the other controlled neurons may. So, it is a long feedback loop, and there are centers in the brain which keep receiving this feedback loop to decide about the further action. So, broadly this was how the brain is formed, and as I said, this as we talk about physiology, a lot of this will become clear to you. So, the basic question still remains, what makes us human? Size of other animals, the brain of other animals is pretty high in some cases. Context, continuity or a break, or connectivity. So, in the next lecture, we will move on to how the brain functions, and then we will, following that, we will look at how do we actually try to peep into the brain. Thank you.